to get you attracted about the things that we are doing in the lab mostly. So what I will tell you today is I'll talk to you about some filamentous pathogens of plants which cause devastating yield losses. So I'll tell you why we need to work on these pathogens. Then I'll talk to you about two of these ones which I have been studying so far. So the main theme of today's talk is about the morphogenesis in these microbes. So they undergo complex developmental changes, and these changes are really important for their infection. So why we study these microbes? Because as you all know, food is limited, and it will be even more limited in near future. We know that the world population will be about 9 billion in 20-30 years, and even now every 1 in 7 person is living undernourished. So we know that food is really important and it's very limited. So as you may have seen these covers from The Economist, so they have said that this is basically a silent tsunami and there is no way to find cheap food anymore. But to me, this is really kind of summarized in this tweet. Sorry for the typo in the medicine, but this tweet says basically medicine can cure you one day, but plants save your life every day. So it is important to study with plants. And one of the serious threats to the plants is these pathogens called filamentous plant pathogens. They can be either oomycetes or they can be fungal organisms. So oomycetes are not fungi, so they are closer to algae, which I'll tell you later on. So these two organisms which have a shape of filaments, they cause devastating yield losses. So what are these ones? For example, they cause blast disease on rice. You may have heard the stem rust UG99, which emerged from Kenya and then now basically devastating our wheat fields. Then we have maize pathogens, potato pathogens, Phytophthora infestans that I will tell you a bit more, and also the soybean rust pathogen. But what I want to point out in this table that was published in Nature a couple of years ago are these numbers. So these are the numbers that these pathogens kill kill the food that would feed these many people. I mean, this is a bit extreme. The second number is a bit extreme. It's almost basically 5 billion people, which would only be possible if all the plants in the world were infected, basically. So I would not believe this one. But this is very realistic. I mean, these pathogens cause about 10-30% of yield loss every year, and this would feed about 600 million people. So this is a very important, serious problem. So how do they cause these massive infections? Because they have huge population sizes. So here, what you are seeing in this picture here, this one is basically the spores. This is not coming from wheat field. These are the spores of the pathogens. As you can see, it is making a orange cloud here. And this is what you get when you walk this boot was normally brown and it's completely orange now due to the pathogen spores. So you have basically enormous population sizes. And this enables them to be highly adaptable. So whatever you do, you can breed for resistance, you can put resistance genes in these plants, but it will not work for very long. So it will work only for a few years and the pathogen will evolve and break the resistance. And they have mixed modes of reproduction, so they can either reproduce sexually or asexually, and this sexual reproduction actually increases their chances of adaptation, and in a very short time, as I said, they will evolve whatever your resistance mechanism is, and they'll cause yield losses again. So we need to understand how the interaction between the plant and the pathogen works, and we need to basically help plants win this battle. So these are some of the pathogens that you may have heard recently in news or in popular news channels. So these are the big ones. So we have rice blast disease, which is causing blast disease on rice. It also infects several other grass species. So this pathogen, for example, gained the ability to infect wheat recently, and it basically devastates the whole wheat fields in Brazil. So it caused 1 year about 80% reduction in wheat yields in Brazil, because none of the wheat plants were resistant to this pathogen. Suddenly it gained the ability to infect a wild grass to the wheat, and basically it wiped out the whole fields. So another one is, this is very recent actually, this black sigatoka disease. You may have seen that our bananas are threatened by this disease very severely, because banana is a basically clonal fruit. So all the clones, all the bananas you have are coming from the same clone. So this pathogen can now infect these clones, and it is basically wiping out the banana fields especially in Africa, which is not the banana that we eat, but the one that is more important for them. And the other ones, for example, potato blight, Asian soybean rust, and wheat stem rust. These are really devastating diseases. I'll tell you about two of them today. So during my PhD, I worked on this rice blast disease. I mainly work on how this pathogen basically undergoes morphogenetic changes to infect plants. And nowadays, I'm working on this potato blight pathogen, Phytophthora infestans, which is the trigger of the Irish potato famine in 1845. So the first part will be on Irish potato famine pathogen. So this is an oomycete pathogen. Oomycetes are closely related to brown algae. So they are not fungi, but they are closely related to brown algae. And this pathogen triggered the Irish potato famine in 1845. As you know, this famine caused death of about 1 million Irish people, and another 2 or 3 million had to migrate to other places. It has been about 200 years since the Great Potato Famine, but this pathogen is still a great problem for us. Every year it kills about $6 billion of potato and tomatoes. So it is still a huge problem. So this is a Phytophthora infested field. This is a resistant line that was bred very recently. As you can see, the potatoes which are not resistant are completely wiped out actually. So I'll just to show you the life cycle of the pathogen, and also it's almost Christmas, so we made a song about the life cycle of the pathogen. I hope you can. Does it work? No? Do we have? It doesn't work?
No, we have an issue in the system, so it won't work. I'm sorry.
Let's try if you will hear like this. The room is not too big, so you may hear. You can hear, right? And the infection is quite quick. I mean, you will see here some potato plants infected with Phytophthora, and you can see within days the plants are completely dead. So this is just another video. It has some music to make it more dramatic even. So. The timelines will come at the end. So these were infected in the greenhouse, and you can see the plants are basically already suffering after a day or so. And this is where the name comes actually. So Phytophthora infestans means plant destroyer in Greek. So this is basically in a week you kill the whole plant, and if it comes to your field, basically the whole field is wiped out in 10 days. So it is a really serious problem. And these beasts are evolving so fast. So if you look at this table, this shows us, so this is coming from a pathogen survey that was done in UK, for example. And so what you see here, for example, if you look at 2003, these different colors are showing different strains of Phytophthora infestans. And you can see they change very quickly. I mean, in a few years, you basically have a completely new virulent strain, which caused a lot of damage. So these pathogens really evolve quickly. And how do they infect plants? So they secrete these molecules called effectors. These are small proteins which basically bind or associate with plant targets. So they are coming from the pathogen, but they function completely as plant proteins. They are quite amazing evolutionary innovations if you think about them. And then they alter plant processes. So for example, they suppress plant immunity, they reshape plant metabolism, or they reshape plant architecture, and then this enables the pathogen to colonize the plant. The resistance happens when some of these effectors trip the wire, and they are recognized by these resistance genes. When they are recognized, they trip the wire. They are recognized by these intracellular immune receptors, and this induces cell death, and this we call effector triggered immunity. You can look at Phytophthora infection from another point of view. So here we have an infected leaf. The red here is Phytophthora infestans, so it is expressing a red fluorescent tag. You can see that this is the infection, this is the initial site of infection. This part is completely dead already, but this part here you have the biotrophic part where the pathogen simply sucks up all the nutrients from the plants and still continues to grow. And in a few hours or days, this part will be dead and the pathogen will continue spreading. So this mechanism, this suction or this subversion of plant processes happens via this special structure called hostorium. So here you have the Phytophthora hyphae, and this penetrates the plants, and from this hostorium you get secretion of these effector molecules. What I want to point out, which I think would be quite useful and critical for you, is this hostorium is when it penetrates the leaf cells, the leaf cells are still intact. So these cells are completely alive. So this is outlined by a host-derived, so plant-derived membrane, and this, even though the pathogen pushes a lot, and sometimes it can occupy up to 80% of the plant cell volume, it somehow keeps these membranes intact. So just to show it in a diagram, so here we have the hyphae, Phytophthora hyphae, or this hostorium is utilized by many other pathogens, not only unique to Phytophthora, but many other pathogens as well. So you have this hyphae here, and then it starts pushing the membrane. It causes an invagination, and somehow this membrane is still intact, so it doesn't rupture. As I said, this part, the pathogen part, can occupy about 80% of this volume. They can stay there for months. They can stay there, let's say for rust fungi, they can stay there for 6 months, but the cells are still completely alive and they don't feel anything. So this is quite an amazing restructuring of the cell membranes and cell metabolism and vesicle trafficking. We have been studying this membrane, this extrahostorial membrane, the membrane surrounding hostorium for some time. We know that they are quite different. They have different composition. So, for example, a protein called synaptotagmin can still, so I should tell you, okay, so these red parts are the hostoria, this digit-like hostoria that you can see infecting the cells. And as you can see here, this synaptotagmin accumulates around these hostoria. But if you look at an immune receptor, let's say in this case we have EFR, these immune receptors are absent at these membranes. So somehow the pathogen made some tricks and gets rid of these immune receptors from this hostoria. But some other proteins such as synaptotagmin is still accumulating around hostoria. So this is another one. So we have also shown that this membrane is not uniform. So it is made up of microdomains, different microdomains, and here we have two different microdomain markers. Again, we have synaptotagmin and we have AVR-BLB2. As you can see that the green and red signal are not overlapping. So basically you have a patchy membrane outlining the infection structure. But what we have discovered recently is this synaptotagmin is actually causing some ER plasma membrane junctions. So synaptotagmins induce basically junction of ER with plasma membrane, and we think this is because there is huge amount of tension at this membrane deformation sites, and synaptotagmin basically supports that membrane site so that the membranes don't rupture. So at the moment we are thinking if we can model this these membrane invagination somehow, and then if we can see what we are thinking biologically is true. So if these invaginations increase membrane tension at these sites, and that our biological model where basically synaptotagmin induced ER plasma membrane junctions can help this pathogen to make these invaginations and accommodation structures. So now I'll stop here because the rest is completely biological, and I don't think it will mean a lot to you, but I want to switch to the rice blast fungus where we actually did some modeling. So rice blast or rice, as you all know, if you go to Asia, this is more or less, a Portuguese writer said, this is equal to life. I mean, rice is equal to life for many Asian people. You grow about 150 million hectares of rice is grown every year. It provides 23% of the world's calories. It's just next to wheat, which is 24 or 25%. So these two staple crops actually provide half of the calories that we get. So most of it is grown and consumed in Asia, and the rice blast disease is a severe, devastating disease for rice. So it causes about 10 to 30% of yield loss every year. It is still a huge problem, and 10% of this yield loss is enough to feed 60 million people. So it is an important disease. It is such a big disease that you can actually see the disease fields from space. So when the astronauts look from space, they can see the Great Wall of China, and next to that you can actually see the disease field. So it is such an important disease. And this pathogen also evolves really quickly, and we have epidemics every 5 to 10 years, and these epidemics cause severe yield losses. So in my previous lab where I did my PhD, we were interested in 3 different questions. So first we wanted to understand how the fungus penetrates leaf surfaces. I'll come to that in a minute. And then when it penetrated the leaf, how does it proliferate within the plant? And then from one cell to another, how does the fungus move? So I'll just go through the life cycle with you because I think it will be important for you to see the morphogenetic changes throughout the life cycle, which are really important for infection. So this is the infection that we study in the lab. So here you see some rice leaves, and here you see speck-like infections. So these infections are, as I said, this is the one which we study in the lab, but they can also infect these necks of panicles of rice plants, and when they infect this, they kill rice leaves, and this is the one that actually causes yield losses in rice fields. So the infection starts with these 3-celled conidia. So here you have an electron microscope image of the conidial cells. So these are 3-celled conidia, and you can see the glue here which helps the conidia to attach to the leaf surface. And then when they attach to the leaf surface, they make this unique infection structure called apressoria. So you will see the polarity changes that's happening throughout apressorium development. So here you have a polarized growth, you have formation of germ tube, then you start growing isotropically and make this balloon-like structure called apressorium. And then this apressorium matures, and you can see when it matures, these parts are completely dead. I'll show you that in a minute. So it requires basically everything in here, everything in the conidia is moved into apressoria. The reason why you need that movement is because it is basically a pressured cell. So here you are seeing a mature apressorium. So within this cell, which is about 8 to 10 microns long, the diameter is 8 to 10 microns. So within this cell, you generate 8 megapascals of turgor pressure. So 8 megapascals of turgor pressure is 30 times more than the pressure within a car tire. You can imagine the pressure within this cell. So the cell walls of this cell apressorium is reinforced by addition of extra melanin layers. How the turgor pressure develops, so the cell basically accumulates lots of glycerol within this cell, and this sucks up water, and melanin layer outside of apressorium prevents diffusion of this glycerol outside, and this is basically a huge turgor pressure bomb. And when it reaches a certain turgor pressure, it ruptures the plant cuticle, and it makes this thin penetration hyphae. So here you have the pressured cell, apressoria, and then it ruptured the rice cuticle, and now you are within the rice cell. So after let's say 24 to 36 hours, this pathogen starts proliferating within the rice cell. So here the pathogen is tagged with green, and here we have the rice cells. And 2 to 3 days later, about 3 days later, I should also add that again, similar to Phytophthora, the fungus is outlined by plant plasma membrane. Again, so the fungus makes these huge infection structures, but during this time, the plasma membrane of the cells are completely intact. Somehow they stay intact. You can see this in here. So this is a time-lapse movie showing the proliferation of the fungus within the plant. So these moving ones are chloroplasts. You can see these cells are completely, well, they look quite happy. They are not dead. So somehow the fungus keeps these cells alive, and then it gets everything for its proliferation. And within a few days, you can see that it completely colonizes rice leaves, and you can see these green parts are all fungal hyphae. This is, I think, about 72 hours later. They start growing through the vascular bundle, and then 4 to 5 days later, you have these infections, which are a centimeter. The diameter is about a centimeter, and from these infections, you get thousands of spores every day, and these basically spread to the field. You can see the life cycle in this cartoon as well. So we start with from conidiophore. So we have a polarized growth stage where we make this germ tube. Then the polarity changes again. So you basically have an isotropic growth stage. And then the polarity changes again. Again, you have a polarized growth, and this continues on within the plant as well. And these polarity changes are really important for this infection. So if you block any stages of these changes, for example, if you block this genetically, you can modify this fungus genetically. So if you knock out the genes that are important for germ tube formation or polarized growth of germ tube, then you will block infection. Similarly, if you block these isotropic growth, again you will block infection. So understanding how these polarity changes happen and how we can actually use this to prevent fungal disease is basically the main research question. What I want to tell you today is a story that I have done during my PhD. So I wanted to understand how this fungus penetrates this leaf surface. So what we knew when I started is this system, this apressorium development is very tightly regulated. So here you have cell cycle regulated control. So what you are seeing here is we all start with this three cell and 3 nuclei. One of them divides, and then one of them goes here, and the other ones are dead. So they undergo autophagic cell death, And this is the same. We have looked at this thousands of times, and we always see the same pattern. So this is very tightly regulated, and it is genetically controlled. And we have already studied the cell cycle control checkpoints of this system. We know that DNA replication is critical. If you block DNA replication, then you won't make this apressuria. So we know that this is a very tightly regulated morphogenetic change. What I wanted to understand is how this break in cellular asymmetry happens. So here you are seeing that apressuria again, this infection structure. So here we have the melanin layer outlining the apressuria. But very interestingly, please remember, so this is a pressurized cell. You have 8 megapascals of pressure here. At the sites where you have leaf surface, we don't have any cell wall. So I was always interested how this cell still stays intact without a cell wall. So, and also how and when the cell knows when to break the cellular asymmetry and penetrate the rice leaves. So as you can see here, during this penetration peg formation, there is a huge membrane tension forming at these sites, at these tension sites, and we were wondering how this is controlled and sensed. So we decided to work with cytoskeleton, and we looked at actin cytoskeleton initially. And when we take the actin with a construct called LifeAct, it's a small actin reporter, you can see that we have a circular structure, a toroidal network at the base of apressuria. So you can see it here a bit more clearly. So you can clearly see that this is like a circle surrounding the apressurian pore. So we wanted to understand how actin network is formed at this pore site. To do this, we started to work on small GTPase proteins called septins. These are considered to be the fourth element of cytoskeleton. You all know intermediary filaments, actin filaments, and microtubules, but these are now considered to be the fourth element of cytoskeleton. And also nice for us, these are not found in plants. You don't have any septins in plants. So these are small GTPases. They were discovered by Lee Hartwell in 70s in yeast. They were discovered for their role in budding. Saccharomyces cerevisiae divides by budding, and these are important at those budding sites. And as you can see here, septins formed a nice ring around apressurian pore as well. And when we do a three-dimensional movie of these rings, you can see that this ring is just at the base of apressuria. So it is where the penetration peg emerges. When we did the colocalization, we can clearly see that this actin network colocalizes with the septin network. You can see this, the actin in red and the septin, and you can see they perfectly colocalize. And as you can imagine from how the story is developing, septins are important for actin network formation. So if you delete these septins, there are 4 of them in rice plus fungus. If you delete these septins, you don't have a toroidal network of actin around apressurian pore anymore. As you can see here, for all of them, SEP3, SEP4, 5, and 6, we don't have this pore anymore. And when we delete these septins, the fungus cannot infect the rice leaves anymore. You can see this is a wild type infection. You have lesions after 5 days, but in the septin deletion mutants, we don't have any lesions anymore. And if you do an electron microscopy in the wild type, you can see after 24 hours, we have the penetration, but the septin mutants, still they land on the plant surface, but they cannot penetrate rice leaves. So after this, what I wanted to understand is what actually induces formation of these septin rings. And when I went and talked with my supervisor, and I said, look, there is huge amount of turgor pressure within this cell. It must have something to do with the formation of these septin rings. And he said, I don't believe you. I don't think it will ever work. But so I started with some simple experiments. So we can develop these infection structures on cover slips. So we can add them on small cover slips, and they will make these infection structures. The advantage of this is we can do basically simple manipulations of turgor pressure. So what we can do is if we add glycerol, I told you these cells accumulate huge amounts of glycerol. If I add glycerol to the outside, what will happen is this will cause a high prosmotic stress, and it will reduce the turgor pressure within that cell. So when I did this, so here we have a normal cell where we just add water. So you can see septins form a nice ring, and also the actins form a nice ring. But when I added glycerol, you can see that I had small rings which cannot, like I lost this ring, and I had smaller rings, which we know that septins have an inherent potential to make these rings. So causing a hyperosmotic stress to the cell resulted in loss of these nice actin rings around a posterior pore. This suggested that turgor pressure actually can have something to do with the formation of these rings. So it can be the inducing signal.
The actin still forms the rings?
No, these are small rings. So you need one ring around the posterior pore. So if you have several rings, because what we know is these rings are telling the cell where to focus the turgor pressure. Because there is no turgor pressure at these membrane wall-less sites. Sorry, there is no cell wall at these sites. So these actin and septins form these rings, and they tell the cell, okay, this is the site where you will push through. If you have several of these rings, then you cannot concentrate the turgor, and it will not be enough to penetrate the cuticle. So it is important that you have a single pore outlining ring. So we check this with genetic mutants. So we can block melanin formation using different mutants. So these are buff and rosy and albino mutants. These mutants cannot form this melanin layer outside of presoria. This means they will not be able to generate this turgor pressure within that cell. And when you look at septin and actin localization within this cell, again you see these small rings which cannot form, and you lose this big ring around the pore. So this suggested that actually turgor pressure is the signal inducing formation of these rings around the presorium pore. Then the question is, if the turgor pressure is the signal, and then if you have these rings forming around the presorium pore for plant penetration, there must be something sensing this turgor pressure. So there must be a sensor which senses the changes in the turgor pressure and tells the cell, okay, now it is time to form these rings and get into the plant cell, get into action. So we know from several other studies that turgor pressure can be sensed in two different ways. So there are two different types of turgor pressure sensors. One of them is mechanosensitive ion channels. So these basically sense the changes in the cell wall, and they induce some signaling mechanisms to adapt to the turgor pressure changes. In rice blast fungus, previously we have deleted all these mechanosensitive ion channels, and they don't cause any defect in plant penetration. So these are unlikely to be the turgor pressure sensor. The other one is an SLN1 two-component pathway. These have been studied in bacteria for a long time. So this pathway is basically a phosphorylase mechanism. So SLN1, this kinase, somehow senses the cell wall changes, the tension on the membrane. And then this induces some phosphorylase mechanisms, and this induces gene changes, gene expression changes, and also some polarity changes. And luckily for us, another group in China already published the mutant of this SLN1 kinase. As you can see, when you knock out this kinase, it becomes non-pathogenic. So here you have the wild type rice blast fungus. You can see it causes lesions. When you delete this kinase, it cannot cause any lesions anymore. And if you complement, if you put this gene back, then you recover pathogenicity. And as you can see here, they cannot penetrate rice leaves. So here we have a wild type cell which is already filled with fungus. Here you have the mutant which cannot, because a posterior couldn't penetrate, so you don't see any fungus inside. And here you have the complemented one. Again, it can colonize rice leaves. And interestingly, when we look at the turgor pressure within these cells in SLN1 mutants, we realize that actually turgor pressure is more than the wild type plants. So this suggests actually, because there is no sensor, the cell continues to build up the turgor pressure, but it cannot, because the sensor is lacking, it cannot induce the repolarization of the cytoskeleton, and you cannot penetrate the rice leaves. And when we localize these SLN1 GFP, we realize that it also localizes around the pore where this whole polarity changes are happening. And you can see this here in a better way. So this is the cell wall autofluorescence, and if you look at the SLN1 GFP, you can see it here in the pore region. And if you, as I said, we can reduce the turgor pressure within apressuria by adding glycerol. And if you add glycerol to SLM1 GFP expressing cells, so here we have the wild type cells, which normally accumulate SLM1 at this pore region. But if you add glycerol, you can see that SLM1 localization is completely disrupted. You don't have polar accumulation of SLM1 at these sites. And also, if you look at these melanin mutants, which cannot accumulate again turgor which cannot generate this turgor pressure, SLM1 accumulation is also again disrupted. So this really tells us that SLM1 somehow is relocalized at these pore sites in a turgor dependent manner, and it induces the cytoskeletal reorganization, which I'll show you in a minute. And what we realized is again consistent with SLM1 mutants, these sensor mutants developing more turgor pressure than the normal ones. They were more melanized than normal apressuria. So these are, if you look at gene changes and melanin levels, you can see that these mutants have more turgor pressure and more melanin levels, but they still cannot penetrate rice leaves. And this is due to defects in these cytoskeletal rings. So here we have again, I'm sure you are now familiar with these rings. So here in a wild type cell, we have these rings forming around opressorium pore. So these are two different types of septins. This is the actin network, and this is an actin binding protein again forming the ring. And if you look at the sensor mutant, these rings cannot form anymore. So if you don't have the sensor, then you don't know how to initiate, how to repolarize your membrane, and you cannot you cannot penetrate rice leaves. So I always wanted to do some modeling with these things, but we don't have much experience on this modeling. So what I did, I emailed Anutida. I'm sure you know him quite well. And I asked him if he can model apressorium penetration, this penetration stage, somehow using mathematical formulas. And he did some modeling. These don't mean a lot to me, but I'm sure it means more to you. So we have different stages of apressorium formation. So initially we have turgor generation, and then we have, just before penetration, we have a different set of formulas, and then these again change during the rupture of the apressuria. As I said, I'm no way close to being an expert on these things, but Anutida is here, so if you want to know more about the details of this thing, you can talk to him. And when they modeled this, so we modeled the components, the four important components. So here we have melanin. So melanin is outlining the turgor, outlining the apressuria in response to turgor changes. When turgor pressure is high, you have melanin in the outline. And when the turgor pressure reaches a certain level, we can see that SLM1 accumulates at the pore. And also SLM1 accumulation somehow triggers repolarization of the septin and actin rings. So you can see when we have the turgor pressure really high, we have these rings forming around opressorium pore. So now this model helps us to come up with some hypotheses, testable hypotheses. For example, if we put another cover slip on top of apressuria, because this model tells us that these rings are forming because they know there is a rigid surface at the bottom. So if we had another rigid surface at the top of apressuria, would we have two different rings forming in the same cell? This is what we are testing at the moment. Okay, so this is the story. So I hope, as I said, I'm quite an outsider to your field, but I hope you are convinced that plant pathogens are a serious threat to our food security, so they are important beasts to work with. They form specialized infection cells, and these infection cells form through changes in their polarity. So these polarity changes can be modeled using different mathematical modeling techniques. And this infection-related development and these morphogenetic changes are critical for infection. So if we can understand these changes, we could actually help plants win this battle and help reducing yield losses due to pathogens. With this, I just want to finish and acknowledge the people that were involved in this work. As I said, I did my PhD in Nick Talbot's lab at University of Exeter. So the rice blast work I presented are from his lab. So I want to thank Nick and also members of his lab. Currently, I'm at the Sainsbury lab working with Sophie and Kamoun. So I want to thank all the members of our lab. And as I said, we are collaborating with Anotida and his team on this rice plus morphogenesis. And thank you for listening.
Okay, are there any questions? Please.
Is there a function of actin in contraction of the actin, or is it just forming a pore?
So it makes a pore at the beginning, and during the so when you have this penetration, then you have this hyphal growth, and this ring very quickly contracts and localizes at the tip of the fungal hyphae and controls the growth.
So the ring is transported.
Yes.
Is following the tip of the growth.
Yes, yes. As soon as you have the penetration, because when you have the penetration, the turgor pressure decreases rapidly. And as soon as the turgor pressure decreases, the highest turgor pressure is at fungal hyphal tip, and the actin localizes at that hyphal tip to basically control that tip, protect that tip from rupturing. So because the fungus needs to push through the plant plasma membrane and continue growing. So at the beginning, until you have the penetration, you have this pore, let's say, protective layer with actin, and then as soon as you have the penetration, you move this pore somehow to the hyphal tip. Yeah.
So you have the fungi resting on the cell wall of your plant, and there will be like an interface.
Yep.
And so that ring, what you see, it's what size does it occupy in relation to that interface?
So it seems, at least in our electron microscopy and also cell biology images, it seems it's almost covering that interface completely.
Okay. So
But there is there is a glue at each side of this ring, and this helps the fungus to attach to the surface. And if you remove this glue from the fungus cannot stick to the plant surface properly.
because your idea about the pressure, the turgor pressure, I mean, it's hydrostatic, so it's everywhere, right? The same.
Yep.
So there's something special about that interface, which is able to spatially find that domain. So my second question is then, in one of your mutants where you had reduced, well, the mutants not, where your treatment, where you reduced the turgor pressure, you still were able to see actin clustering, but then in very small domains.
Yep.
And so is that, were those small interfaces, or was it one huge interface but then small clusters of actin that you got?
So it seems there is still one interface,
Yeah.
but these actins, so they have inherent mechanisms to make these clusters, because septins can somehow scaffold them to different clusters, so they still make these clusters. So there is still one interface. When the fungus sticks to the plant surface, actually, there is no melanin layer at this site. So we think because there is no melanin layer, that kind of acts like a glue as well, because it pushes so hard, then you somehow stick to this place and you don't have any melanin layer at that site. So then this helps the fungus to know where to make this penetration peg, actually. But actins, if you deform them somehow by reducing the turgor pressure, they make these rings inherently at some other size. Any more?
I had a question with the first pathogen where you had the Haustorium.
Yep.
You had this very long membrane, or large membrane, and you were saying it remains intact.
Yep.
You said there were possibly high strains and high tensions.
Yes. Well, this is
what
what we
is your
think.
evidence for that?
So-
or is there a growth of the membrane?
Yes, the membrane continues to grow. So, I mean, as I said, the evidence is coming from biology, because we know that these proteins form ER plasma membrane junctions, and these ER endoplasmic reticulum plasma membrane junctions, and they have been shown to be involved in helping this membrane tension, like, let's say, buffering this membrane tension. That is our evidence. So we never measured the membrane tension, for example. But what is kind of common in all pathogens is they induce these deformations in the membrane, and somehow these membranes stay intact as long as the pathogen wants them to stay intact. So we think this, for example, this synaptotagmin may be one way that the pathogen actually triggers formation of these, let's say, support sites, and that is how it keeps the membrane intact. But it would be great if there is a way to actually somehow model and measure the tension at these imagination sites and kind of back this up with the biological data we have.
Question is that how fast is that haustorium growing?
So you have the haustorium formation, let's say within 24 hours, the haustorium already forms.
Okay, then I would expect that the lipid flow in the cell would be pretty fast, fast enough to supply the needed additional lipids to the plasma membrane. And then in the end, the plasma membrane is just a two-dimensional liquid, and you have normal pressure onto it, but you wouldn't have an extensional tension that would be enormous. So the lipid structure itself reacts to pressure. You increase density, and this might be an effect for some of the proteins, but and the mixing and the formation of domains, but you wouldn't have classical membrane tension in high to some high degree.
Okay.
That's what I would expect. It's science fiction.
Thank you. Any more questions? Okay, well, let's thank both speakers again, and then we can break for coffee, and I think we return at 11:30.